Welcome to computational genome biology. For decades, the goal of oncology was to sequence a tumor's DNA to find every answer to cancer. But even with a complete genetic map, our understanding of why some tumors survive treatment remained incomplete. DNA provides a map of potential mutations. However, it cannot capture the functional execution of a tumor, such as post-translational modifications or the metabolic states that actually drive disease progression. Think of DNA as an architectural blueprint. If a building collapses, studying the floor plan might show you where a support beam was supposed to be, but it won't tell you if the actual steel was compromised. You have to inspect the physical materials. In biology, those physical materials exist in deeper layers. We have the transcriptome, which represents the RNA instructions, and the proteome, the actual physical proteins performing the work inside the cell. When we integrate these layers, we enter the field of multi-omics. This approach captures the active protein networks and signaling cascades that define a tumor's actual biological behavior. Genetic sequencing successfully identifies primary driver mutations, but observing how that code is physically executed through proteins provides the precise foundation needed for risk stratification and target selection. To solve this integration problem, the National Cancer Institute established the Clinical Proteomic Tumor Analysis Consortium, or CPTAC. CPTAC uses a method called Proteogenomics. They take the exact same tumor sample and analyze it across multiple biological layers, utilizing high-resolution mass spectrometry to identify and quantify the proteins inside. The raw DNA and RNA sequences extracted from these physical samples are harmonized, formatted to a single standard, and stored in the Genomic data commons. As this flowchart shows, the corresponding mass spectrometry data is uniformly processed and housed separately in the Proteomic data commons. These portals form a centralized, open-access ecosystem. They host terabytes of linked data, completely free for researchers across the globe to download and study. CPTAC provides the foundational public infrastructure required to give any scientist on Earth a complete multi-omics picture of the samples they study. This infrastructure solves concrete clinical problems. Consider the difficulty doctors face when diagnosing thyroid nodules, where it is incredibly challenging to distinguish a benign adenoma from a highly invasive carcinoma. Traditional visual biopsies and pure genetic inspections frequently fail to tell these specific thyroid nodules apart, leaving doctors and patients guessing. To solve this diagnostic failure, researchers are pulling harmonized multi-omics data directly from the CPTAC portals and using it to train machine learning algorithms. This scatterplot visualizes how algorithms like the random forest classifier scan massive proteomic datasets. The algorithm processes thousands of data points to identify subtle protein signatures that human eyes miss. The algorithm successfully isolated specific proteins, such as C2C2L, and variations in thyroglobulin levels that perfectly separate malignant from benign conditions. Open-source multi-omics data, combined with artificial intelligence, enables researchers to pinpoint life-saving diagnostic markers that remain invisible to traditional medicine. Science is currently making the leap from the initial genomic era into the far more complex post-genomic era. Precision medicine is no longer constrained to hunting down a single primary driver mutation in a patient's DNA. The emerging standard of care requires mapping the entire active protein network of a disease to fully understand its physical behavior and execution. Resources like CPTAC and the Proteomic data commons are actively closing the genotype-phenotype gap, linking the DNA blueprint to the physical building. With this open data, researchers worldwide can build precise prognostic models and uncover therapeutic vulnerabilities that we couldn't access even 10 years ago. By seeing the complete multi-omics picture of cancer, humanity is building the tools to stop the disease before it adapts. Thank you for watching, and please like and subscribe to the Computational Genome Biology YouTube channel for more.